So the final exam, let's go through kind of the logistics of it. I feel purple today. So for the lecture component, there's going to be 100 questions. Of those 100 questions, 50 of them will be on the old materials. And old materials is anything from exam 1 to 3. And then the other 50 is on the new material. So you can technically think of that as exam 4. Okay, so with that 50 questions on the old material, I've probably covered about 15 chapters in exam 1 through 3. So that's going to be maybe about like 3 to 4 questions per old chapter. So with the old materials, I would just kind of briefly review them, but I would focus on the new material, to be honest. So the new material, which is on urinary, endo, reproductive, that is divided within that 50 questions. Now within the 50 questions, 40 of them are multiple choice and 10 of them are fill-ins, just like, you know, the first 3 exams you had taken. Any questions on this?
Professor, are there any like short answer? Because I saw on the email that there might be some short answer questions.
Yep, the fill in the answers are the short answers.
Okay, thank you.
Yep. Will there be fill in the blank? Yes. Oh no. Oh, so Brianna asked, will there be fill in the blank over the old materials? And that's a no. So the 50 old material are all multiple choice.
That's my question. Thank you.
Yep.
So
Other
only
questions?
10 out of the 100, 90 of them are going to be multiple choice. What we're saying.
Say that one more time.
90, not it's Jamie. 90 questions are multiple choice. 10 are fill in the blank.
Correct. That is correct. Yep. Other questions? Okay, so really if I were a student, I would focus on the new material. And then as you're studying the new material, you know, look back at the old material, and you should find that the old material is like, oh, I knew that, or oh, I know that, you know, but still review the old materials. I'm not saying don't review it. Do review it. A good review source would be all your old quizzes and all your old exams. You should be able to see all of them. Okay, not the correct answer, but you'll see your answer and whether you got it right or wrong. Okay, so that's a good review, just to kind of go through all your old materials, your old study materials, your old quizzes and old exams. Excuse me. And then make sure though you really do focus on that new material. Okay, so that's for lecture. And then for lab has 50 questions. So about 25 will be on the old material. And about 25 will be on the new. And they're all the same type of questions. They're all identified, and you have to type it in spelling counts. Type in, and then spelling counts. Pay particular attention to if there's a pair of something, make sure you indicate whether it's right or left. If they're, like, for example, with blood vessels, make sure you identify whether they're an artery or a vein. You know, make sure you have all the parts to the names to them. Okay. Let's see what else.
Professor, can I ask a question? This is Jamie.
Yes.
Listen, what if there's a image that has the arrow pointing at it, and you're looking at it, you go, okay, like for an example, okay, it's the liver was the answer, but I put left lobe of the liver, and so you know, how do we, you know, and I don't, if there's a question or there's a doubt in our mind, is it okay for us to say, well, it's either this or this? I don't know which one you're pointing to. You know, it's because we can't ask a question during the exam.
Right, right. So I will only take one answer, unfortunately. So if you put multiple answers, multiple different answers, I cannot grade that even. So I can only grade it if you put one answer. But if you're ever in doubt, that one example you gave me, for example, it's like this, right? And the arrows here. It will always say it. Always go with the specific.
Okay.
And what you said was perfect. So for example, you said the right lobe of the liver. And you see, when you write it like that way, you cover, if I were to ask it generically what organ it is, as well as specifically what it is. So either way,
Okay,
you get it right.
Covers your bases there on that one.
It covers your bases, exactly. So that's a great example, Jamie. So I hope everyone followed that. So if it's something, if I'm asking, if you're not sure whether I'm asking for that specific thing that I made you know on the PDF or the overall organ, put in both. Right lobe of the lung, right lobe of the liver. Now here's the problem though. If you put in, let's say for this, I'm asking for this specific area right here, and you put right lobe, but then you put of the lung, you will get that wrong. Okay, so you have to be really careful. If it's the liver, put it the liver. If it's the lung, right lung. Okay. Other questions?
I was just going to add, Professor, that to Jamie's question. What I do is I just focus on the question itself. Like if it says what organ, what structure, what vessel, that usually helps me narrow down the specific type.
Exactly. So that's perfect, Aaron. So if you read the question carefully, it will actually tell you what chamber is this, what blood vessel is this. It will say, or what organ is this, or hint, if it's pointing to the haustra, it'll say hint, what curves are these, and so on. So read the questions. Yep, for sure. That's a great recommendation. Okay. Let's see what else. Yeah.
It kind of doesn't go into the review, but just a general question. For like once final grades are in, what is your rule on like rounding up? Like let's say if your grade is like, you're like 0.5 away from the next letter grade, what is your rule on that?
I will always do it in the benefit of the student. So if they have like 72.5 or like 72.6, so on upwards, I use the 5 and upwards. Then you'll end up with a 73%.
Thank you.
Yep, great question. Yeah, I always round up. I actually just tell the computer to automatically round up. Yeah, other questions? Okay, so I need to go through the other portion now. So you know that you need to have respondus lockdown. And more important than ever, because with the final, I normally, if respondus flags you, then I watch it, but for the final, I have to watch all of them. So make sure you do an appropriate environmental scan. Okay, and I gave you a video of what one looks like, so if you're just showing me like up and down, that, I can't tell, so you have to show me like your workspace to your right, to your left. So watch the video. If you are in doubt of what I am expecting, watch the video of what a proper environmental scan looks like. And unfortunately, I have to watch all of them, where before I just waited until I was flagged. Okay, so make sure you do a proper environmental scan. The webcam is on. So that's the logistics: respondus lockdown, appropriate environmental scan, webcam. Please don't wait until the last minute, and you'll find that unlike exam 1, 2, and 3, the dates for these exams are Friday to Sunday, not until Monday, okay? You do not have until Monday to take this exam. And the reason for that is because the last day of class is Sunday. So just to let you know that you only have until 11 p.m. Central Standard Time on Sunday to take the exam. Okay, so Sunday, okay, not Monday, Sunday. Okay. And then what else? Someone asked when I'll get the grades up. Usually I try to get them up by Monday or Tuesday. And that will give you a few days to go through your grades and check them, everything. And then I have to submit by Thursday. So if you're going to check your grades, check them by Thursday, because that's when I have to submit grades by.
Professor, real
Yes.
fast.
Yeah.
When do we have access to the exam on Friday? Is that midnight on Thursday night your time, or around that time, or is it midnight Friday night, Friday, or is it what time?
I think it's midnight Friday. So like
midnight,
I think 11:59 p.m. Thursday.
Thursday, exactly. That's exactly right. Okay, thank you.
Mm-hmm. Okay, that's a great question though. Other questions, and I'll double check too. Other questions about the logistics or just anything? Don't get nervous. Think of it as just like a fourth exam. Give yourself enough time. Read the questions carefully. Have some food by you. You know, there's nothing wrong with having food around you. Have whatever, you know, you need to give yourself energy. All right. Okay. So the 50 in this review session, I'm going to cover the urinary, a little bit of the endocrine if we have time, and then the repro will be for this evening. Okay. So let's get started. If there's any question, are we still? Absolutely. So Allison asked, are you still allowed to have a clean sheet paper? You can have multiple clean sheets of paper as long as you show them on your environmental scan, front and back, that there's nothing on them. And then at the end, once you're done, show the front and back again. Okay. So yes, you are absolutely allowed to have clean pieces of paper. Okay. All right. So let's get started. So for the urinary system, the overall kind of what you need to know for this is make sure you know the functions of the kidneys. For example, filtering blood is a really big one. And in filtering blood, it not only filters it like the way we think it does, you know, like it doesn't just remove and separate the waste, but it also has to bring back the good stuff. Okay. So by filtering blood, not only do you separate the good stuff from the bad stuff, but you also have to return the good stuff. Okay. So that's when we'll start talking about the filtration process, about how there's filtration, but there's also reabsorption, secretion, and so on. Okay. So two parts to filtering blood. One is going to separate the good from the bad and then get rid of the bad, but then bring back the good. Okay. So by doing that, it's also going to regulate water volumes. If it's regulating water volumes, it's also going to regulate pH. It's also going to regulate blood pressure. It's also going to regulate, you know, anything that's related to water volumes in blood. So those are the major functions of the kidneys. In addition to that, it actually also produces hormones. What hormones does it produce? You know this from the previous chapter. From bone or red blood cells, actually. Do you remember hypoxemia? If you have hypoxemia, the kidney senses it and produces this hormone that then stimulates the bone marrow to produce red blood cells.
Is it calcitonin?
Not calcitonin, but good guess. Do you remember a few you did the hypoxemia video? What was secreted by the kidneys to increase red blood cell production? sometimes athletes dope on this, like it was a big thing with Lance Armstrong and the bikers. Erythropoietin, EPO for short. Erythropoietin is a hormone that is produced by the kidneys. It causes, it leads to red blood cell production. Also renin. We didn't really focus on renin that much because it's more of AMP2, but it helps to regulate blood pressure, water volumes and such, okay? All right, so 3 main things that the kidneys does: filters blood, regulates water volume / pH, and then produces hormones like EPO and renin. Make sure you can describe the gross anatomy of a kidney. That's the structures that you can see with your eye, which includes the fibrous capsule. The hilum is just a space that things enter and exit. The associated blood vessels, the renal cortex, the medulla, the minor and major calyxes, and then the renal pelvis. Okay. Then in terms of microscopic anatomy, make sure you know the nephron. These are the functional units of a kidney. Okay. So the nephron is actually what does all that work in terms of like filtering, regulating water volumes and such. So we consider the nephron to be the functional unit of the of the kidney. Functional unit of kidney. So the nephron is the functional unit of kidney. Make sure you know the parts, the glomerulus, which the glomerulus technically is not part of the nephron, but the glomerular capsule surrounds the glomerulus. And then you have the renal tubules, which include the PCT, proximal convolute tubule, nephron loop, DCT. And your book does not consider the collecting duct as part of the nephron, but it is connected to it. So that's why I added it there. Make sure you know the structure, the function, and the tissue linings, which is what we'll focus on at a request. So we'll be looking at that. Know the urine formation process. That was also a request, so I'll be focusing on that. And then know what continues after the kidneys, which would be the ureter, the urinary bladder, the urethra. Know the structure, function, and layers, and then be able to describe the micturition process. Okay. All right, so we'll go right into it. Gross anatomy, just do a quick quiz. What's number 2?
The left kidney.
Left kidney.
Very good, left kidney. Number 1?
Left adrenal gland.
Yes, I love how you're using your rights and lefts, okay? How about number 4?
Urinary bladder.
How about number 9?
Right adrenal gland?
No, it's pointing to the red tube.
Oh, I didn't see that downward. That is the right artery.
Renal.
Yes, it's the right renal artery. Yep, great. How about number 3?
Left ureter.
Great. So you got the gross anatomy. Pretty straightforward of the urinary system. Okay. So this is what we'll do the gross anatomy of kidney. Sorry, it's so blurry. I don't know why it's so blurry, but you know what? I'll just draw my own. Okay. All right. So you can see that the kidney has, if we were to look at it with layers, the outer protective layer is called what again?
Fibrous capsule.
I'm so sorry. I must have like hard of hearing, but can you speak up?
Sure. Sorry, I was away from my mic. The fibrous capsule.
Yep, exactly. And I'm gonna plug my computer in before it runs out of battery. I don't want that to happen. Okay. So number 1 is the fibrous capsule, great. And this layer right here what would you consider that as?
Renal cortex?
Excellent, the renal cortex. And then everything here,
Renal medulla.
perfect. Now in the renal medulla, you will have these slices of pizza because everything is about food for me here. So in the renal medulla, it consists of two main parts. What are the two main parts it's consisting of?
The renal pyramid.
Yep, and
Renal cortex.
The renal column?
the renal column is correct. Thank you. Now you'll notice that I'm drawing these straw-like lines through the renal pyramids. That's going to represent what you're going to learn later to be the collecting duct. Okay, so 3 would be the renal medulla. Within 3, you have 3A, 3A, and 3B. 3A is the renal pyramid, 3B is the renal columns. Okay? Questions? Okay, then at the end of each renal pyramids, there's kind of like a little cup that collects whatever is produced. Okay, so this portion right here, at the end of each renal pyramid, there's an individual cup. What is that structure called?
Minor calyx.
That's the minor calyx. When you have 2 or more minor calyx come together, like right here, then you have number 5, which is the,
Major calyx.
yep. When they all join together, like right here, then it becomes number 6, which is the,
Renal hilum.
which one?
The renal.
Renal hilum.
The
yeah.
renal
No.
hilum is a space.
The pelvis?
Yes, the renal pelvis. Excellent. Thank you. And then finally, when it exits, it becomes this whole tube right here, which is number 7.
Ureter.
Perfect. Okay. So just to kind of make sure we know this, let's do yellow actually. Okay. So this space right here, so it's not an actual structure, it's a space where things can exit and enter, where there's no fibrous capsule. That's the renal hilum. Okay. All right. So that's the macroscopic anatomy. Now, just to give you a placement, the functional unit of a kidney looks something like this. Okay, I'm going to zoom in so you can see. That's to show you one nephron. Okay, so the nephron you can see lies in what layer?
Cortex.
Yep, it can be solely in the cortex, and part of it goes through there. Okay, so that's the collecting duct. As it moves through the renal pyramids, that it becomes a collecting duct. So you can see that you can have different types of nephrons, but the majority of them are majorly in the cortex. You can have some more deeper down in the medulla, but that's what it looks like. Okay, so now that you kind of know the placement of it, we're going to spread it apart, and it's going to look something like this. Okay, it's not going to look clean like this. Okay, this image right here has been kind of stretched out so you can see in its individual part. Oftentimes it's overlapping, it's like switching upside down, it's crazy. You just kind of have to follow the path, the flow, because it only flows in one direction. But this is not how you would find it in your kidney, is what I'm saying, you know, because this is nice and stretched out so that you can see the individual parts. Okay, so let's start. So with the microscopic anatomy, in other words, we're going to be looking at the nephron. Let's start with this first part right here. What would number 1 be?
Would that be the renal corpuscle? Is that what that? or no?
Yes and no.
That's why
So,
I'm a little confused with how they label it. So yeah, if you could kind of flush that out for me.
yeah. So now if we're looking at just the nephron,
Okay.
what would number 1 be?
Glomerular.
Yes. So number 1 is the glomerular capsule. Okay.
Gotcha.
So the glomerular capsule. Now the glomerular capsule, I'll just write out. I normally just number it so that you can use this as a study guide, but I'll just write it out. Glomerular capsule. If you went to school a long time ago, we used to learn it as the Bowman's capsule, but we're moving away from names. Okay, so this is the glomerular capsule. So the glomerular capsule is the first part of the nephron. Okay, what is it lined with? What is the glomerular capsule lined with?
Simple cuboidal.
Perfect, thank you. Simple cuboidal epithelium. That's the tissue name. And then we can even go even further and say the simple cuboidal epithelium are made up of what type of cells? Just like in the digestive system, along the gastric pits, you have the simple cuboidal epithelium as well, right? But they're called absorptive cells. Here you have simple cuboidal epithelium. What cells would you find here? They look like little feet.
Not podocytes.
Yes, yep,
Oh,
that is
okay.
them, the podocytes. Okay, so the glomerular capsule is like a baseball mitt. You know, you can stick your hand in it, you know? So the capsule has 2 layers, right? So the capsule looks something like this. It's got this outer layer, the parietal, and it has this inner layer, the visceral. Okay, so they're both lined with simple cuboidal epithelium, but the visceral layer is going to consist of podocytes. And the podocytes look something like this. They look like little feet with these little toes or extensions. And then there would be another one over here. And you can see that they form kind of an interlocking structure, or what we call the filtration slits. So the podocytes look like these little feet with toes. And then when the toes kind of interlock with each other, they form what's called filtration slits. And that's important because that's going to help allow only certain things to be filtered and keep certain things in the bloodstream. Okay. All right. So those are the podocytes. Okay. Questions? Okay. All right. So for now, we're only going to look at structure. Next time, once we're done with this, I'll come back to the same image and we're going to add function. That's when we're going to talk about the filtration process. Okay. But for now, I just want to stick with structure. Okay. So that's the glomerular capsule. It's got the simple cuboidal epithelium, podocytes, and so on. And this is going to be the site of filtration. Okay, so a site of filtration. Then this component right here, that's going to be number 2. What's number 2?
That's the proximal convoluted tubule.
Perfect. Proximal because it's the closest to the glomerular capsule. convoluted because the tube is curved, and tubule because it's a tube. Okay, so it's the PCT for short, PCT, proximal convoluted tubule. Okay, the proximal convoluted tubule is going to be lined with what?
Simple cuboidal epithelial tissue.
Simple cuboidal epithelium. But what's specialized about it?
It has the microvilli.
Perfect. And what do you know about microvilli? Whenever you have
It increases
microvilli,
absorption.
yeah, exactly. It's for exchange, right? For absorption, right?
Absorption.
So knowing that it has the microvilli, you know that there's going to be a lot of movement of things here. Movement of substances. Okay. All right. So it turns out that it's going to allow for the majority of reabsorption to occur here. And again, I'll come back and I'll talk a little bit more about the reabsorption. For now, just kind of know that this is where the majority of reabsorption occurs. So microvilli allows for absorption. Why are we calling this reabsorption? Why are we calling this process reabsorption?
Because there was already some some of the reabsorption that was taken during the glomerulus.
No. So reabsorption is the fluid going back into the blood vessel.
Right.
So that didn't happen in the glomerulus. Glomerulus and glomerular
Oh, that was the
capsule only
filter.
allow for filtration.
That's right. That's right.
So do you remember the digestive system? So in the digestive system, the nutrients were brought into the blood vessel, right? So that's the first time it happened, and that's why it's called absorption. This is the second time it's being brought back into the blood vessel, and that's why it's called reabsorption, because you absorbed it the first time already in the digestive system. So that's why this process is called reabsorption, okay? Because you absorbed it once through the digestive system already. Okay, so that's the proximal convoluted tubule. Simple cuboidal epithelium with microvilli, so that means that there's movements of substances, okay? Then this bottom portion right here, this entire bottom portion is referred to as what?
The nephron loop.
Yes. Nephron loop. If you studied this a long time ago, it used to be called the loop of Henle, okay? So the nephron loop. Now the nephron loop does have two portions. (3A) and 3B. What's 3A?
Descending and ascending.
That's right. The descending. And don't forget what term, otherwise you'll get 50% off or 0.5 off.
Yeah, to nephron loop.
Descending
Descending
limb.
nephron loop.
Nope, you gotta have the limb, okay? So descending limb, and then if you want to clarify, of the nephron loop.
Gotcha.
Yeah. Okay, where this then is the ascending limb. Of nephron loop. Now you can notice that the ascending limb is mostly the same type of tube, right? It's this thicker tube. See the thickness of it? Okay, so it's basically one type of tissue. Where the descending limb has two parts, right? It has this thickness here, and it's got this thickness here. Notice the difference, okay? So you know that if their structure is determined by what? Right? Remember the one thing I said, I want you to leave this class with is that you should know that structure is determined by what?
What it does.
Yes, function, exactly. So it's structured differently because you know it has different function. And because of that, you will need to know its function, okay? So the ascending limb is pretty straightforward, right? But the descending limb has two segments, the thick and the thin. So we need to also go through that. So the thick segment and the thin segment. Why? Because structure is different, which means function is different. Okay, so the thick segment is going to be lined with what?
Is it stratified? Tony?
Nope, it's still simple. Simple, cuboidal with microvilli, where the thin segment is simple what?
Squamous.
Perfect. Epithelium. Okay, and the reason for the difference in shape is because, again, we'll go through this in a little bit more detail, but the thick segment with the simple cuboidal with microvilli, it's going to allow for the reabsorption of a lot of things, not as much as the proximal convolute tubule, but still a lot of things. By the time it gets to that thin segment, simple squamous epithelium, it's usually only going to allow the movement of water, okay, because it's thinner. So, you know, with the simple squamous, it allows for the movement of water more easily than it would other substances infiltrate. Okay, does that make sense? Okay, so the simple squamous, you can think of it as it's permeable to what?
Water.
Yep. So you would
Also,
expect,
sorry. Also,
No, go ahead.
I just wanted to add that on the thick segment, I have it on my notes that they have a lot of mitochondria to produce ATP, whereas in thick segment, they don't have as much mitochondria.
That's right. Because when you have the movement of water, it's through osmosis, right? And does osmosis require energy?
Yes.
No,
It
it
does
doesn't.
not.
Oh,
Osmosis is passive movement, so it does not require energy. Where in the thick segment, it's responsible for moving a lot of the electrolytes, the salts. Okay, and with that, you have to have the potassium sodium pumps. Okay, and whenever you hear pumps, what do you think? Passive or active?
Active.
Yeah. So that means it's gotta have it relies on active transport. And if it relies on active transport, there must be mitochondria. And since we're doing the thick segment of the descending limb, we might as well go back and do the proximal convoluted. In the proximal convolute, the majority of reabsorption happens here, which means the majority of movement of things happening, okay? So that means that it's definitely going to be active transport. And if it's active transport, it will also have lots of what? Mitochondria.
Mitochondria.
Yep. Okay. That's a good point. Thank you for mentioning that. Okay. So thick segment, Lots of mitochondria, active transport. It's moving the electrolytes in the salts. Thin segments, the squamous, permeable to water. Water moves through osmosis. So that means that there's no mitochondria needed here, right? Because there's no ATP needed. When you go back to the ascending limb of the nephron loop, okay, there's less movement of things happening here, just overall less movement. Because everything has already been moved through either the proximal convoluted tubule or the descending limb. So by the time it gets to the ascending limb, there's just less movement of substances. Excuse me. Less movement of substances. Okay. All right. And then by the time you get to this portion right here, oh, we didn't say what it's lined with. That's, sorry. The ascending limb is lined with what?
Would this be simple cuboidal, but there's no microvilli in there?
Less, not none, but
yes,
less, definitely.
Less, okay.
Simple cuboidal. Happy with less microvilli. Okay, so definitely less, which makes sense because at that point there's less reabsorption, less movement overall. Okay. All right. Okay, so then this next part right here, what would this be then?
Distal proximal tubule.
Yeah.
I mean, distal.
Distal.
Wait,
Distal convoluted tubule.
There you
convoluted.
go. So I'm going to put that at 4. So that's the DCT, the distal convoluted tubule. Now you can notice it's convoluted, so it's curved. It's distal because it's further from the glomerular capsule. So that's why it's called distal, and it's a tube, so it's tubule, okay? The DCT, the distal convoluted tubule. What will this be lined with?
Cuboidal epithelial tissue, but does not have microvilli.
Yes. Okay. So my pen ran out of energy, so I'm using my mouse pad. So I'm gonna see if I can type it in there. Insert home. Yeah, totally not digging this new computer. It runs out of battery so much faster, even though it's newer. All right, I'm just gonna type. I'm gonna write it in there. So simple. Simple cuboidal epi, and then without microvilli, because at this point there's just not much moving anymore. Okay. And then we'll get to this portion right here, which is number 5. What is that?
Collecting duct.
Mm-hmm. Okay. And technically the collecting duct is not a part of the nephron. So we'll kind of just leave it as the collecting duct. Although few things like some salts, very little salts, but mostly water can move here. Okay. All right. So that's pretty much it for the
Sarah,
nephron. Yeah.
Can I ask one quick question? So just to make sure all the main parts of the nephron, can you just go through that really quickly? Because I believe you said that the glomerulus is not part of the nephron, unless I misheard you.
No, that's correct.
Okay, could you go through that really quick? Just like what are the components of the actual nephron?
Yeah. So the nephron consists of number 1 through number 4. So the glomerular capsule, the nephron loop, the PCT, and the DCT. So those 4 parts.
Gotcha. Thank you.
Yep. And you know, so that's a good point. Let me just kind of highlight them. There. Okay. All right. So now I'm going to come back to this image and we're going to add on the blood vessels. Let me see what the next one is. So here is the same image again. But now we're going to add on the blood vessels. Okay. So you can imagine that this blood vessel will go out like this. Now, I'm not requiring you to know all of the transitions of how it started out as the renal artery, becomes the segmental arteries, interlobal arteries, arcuate arteries, cortical radiate arteries, and so on. I just want you to know that it starts out as the renal artery. It enters the kidney as the renal artery, and it transitions until eventually it becomes this. And what is B?
Is B afferent arterioles?
Absolutely. And if you remember from the blood vessel chapter, an arterial is a smaller artery, right? So you've got artery and then becomes an arterial. Now, once it becomes an arterial, it's going to be surrounded by the glomerular capsule, but it's a network of capillaries like this. It would look something like this. Okay, so in here, you have C. What would C be? The glomerulus, exactly. It's very similar to like a ball of yarn. Think of it as a ball of yarn, but essentially made up of blood vessels, capillaries. Okay, so this would then be the glomerulus. And the glomerulus is a network of capillaries. Makes sense because it goes from artery, arterial to capillary. Okay, and we know that capillaries where exchange occurs. Okay, now then it's going to leave and become this blood vessel right here. And then this, but let me make this a little bit thicker so I don't have to keep tracing over it. Okay, so what would this be right here?
Afferent arterial?
Yep. At this point, you're like, what? That's kind of weird, because remember when we studied the blood vessels, it was artery to arterial to smaller versions, you know, but then eventually became capillary. And then after capillary, it became what? Veins. Okay, so this is the normal transition of your blood vessels, artery to capillary to veins, except for like 2%. This is the renal arteries are the one of the 2% where it goes from artery to capillary back to being an artery or an arterial. Okay, and another thing to help you remember, A in afferent is arriving. E in efferent is exiting, just to kind of help you remember the flow of it. So it goes from renal artery to A as an arriving arterial to the glomerulus, and then it becomes the afferent, and then it becomes the efferent arterial, E for exiting. Then the efferent arterial wraps itself around the tubes, the renal tubules, right? The PCT, the nephron loop, the DCT. When it wraps itself around like that, it's called the what?
The paratubular capillaries.
That's right. Para meaning around, tubular because it's wrapping around the tube, capillary because it's going to allow for what to happen?
Exchange.
Yeah, makes sense, because what happens along the renal tubules? Reabsorption and secretion. So wherever you see capillary, here's a capillary, some exchange happened there. What do we call that exchange? That happens between the glomerulus and the glomerular capsule. What's the first step in urine formation?
Filtration?
Yeah, thank you. And here's another capillary. That means exchange occurs here again. So exchange occurs between the renal tubules, all of them, and the paratubular capillaries. And what two processes happened here?
I'm sorry, you said between the tubules, like the descending limb and
Mhm.
those, and the paratubular capillaries?
Yes,
That's excretion and reabsorption?
not excretion, but
Secretion.
there you go.
There you go, sorry.
Nope, that's good. Okay, all right. So that's kind of what happens at the capillaries. So the paratubular capillaries, here's a note I need to make though. The paratubular capillaries, if it's in the renal cortex, is called the paratubular capillaries. But if it's in the renal medulla, it actually goes by another name. Does anyone know what that name is? The vasa recta. Okay, but essentially it's the paratubular capillaries, just in a different location. Okay, all right. So then the paratubular capillaries wrap and then leave, and when they leave, they're called what? D. They're going to transition, but when they leave the kidney, it's the renal vein. Okay. All right. So we covered the parts of the nephron, 1 through 4. We covered the blood vessel that wraps around it, A through D. Okay. Questions? Okay. All right. So now we're going to kind of review what this is a really good figure to have. It's figure 23.22 in your textbook. Okay, it's a nice one because it shows you exactly what's happening. Okay, so it doesn't show you filtration, but it does show you reabsorption and secretion. Okay, so you can see that the majority of reabsorption happens where?
In the PCT?
That's right. So all the things that is in plasma is going to be filtered into the glomerular capsule. So that's filtration. Okay. Then of course you have to separate the good from the bad, but you also want the good stuff back, right? Because in glomerular filtrate rate, you filter like 150 to 180 liters per day. Liters per day. If you lose everything that you filtered, you'd be in the bathroom all the time, you'd dehydrate and you'd die. So clearly we want back like 80 to 90% of what we filter. And that's why reabsorption is really important. So you can see that the majority of reabsorption occurs at the PCT. Okay. Then you have water being absorbed. Water being reabsorbed here. Okay. You have salts being reabsorbed here. You have minor salts being reabsorbed at the DCT. And you have water and a little bit of urea being reabsorbed at the collecting duct. Okay, so you can see that thin segments is always going to allow for reabsorption of water. Why? It's lined with simple squamous epithelium. Where the thick segments are going to allow for reabsorption of the salts. Why? Because it's going to be lined with the simple cuboidal epithelium. Okay. All right. Then you do have secretion happening. Okay. So with secretion, it's going the opposite way. It's going from the blood vessel into the renal tubules. Okay. Why would you want that to happen? Why wouldn't it just all go in this first step right here, which is filtration? If you already have filtration, which allows you to go from plasma to filtrate, why would you want secretion right here, which is also going from plasma into becoming filtrate? Why? Why do you need two? Why can't you just have filtration?
Isn't it because some of the stuff leaks out and not all of it does, like, gets into the filtration process, so it has to go through it again?
Could be. Yep. So I use the analogy of like washing your clothes. Really, your kidney is like your clothes washer. You throw dirty clothes in there, and the washer separates the dirt from the clothes, gives you back your clothes, gets rid of the dirt. But let's say, and I do this all the time, I put all my laundry in there and I realize I found a sock here, I found a shirt there. I'm like, oh no, do I have to do a whole new load again? Or do I just toss them in last minute? What do you think I did?
Toss them in last minute.
I toss them in last minute. Exactly. Okay. So really filtration is the main way of getting blood clean. But let's say sometimes, you're all suddenly you just drank like five gallons of water, and now you have too much water that you didn't get rid of through filtration. So you still have to have some way of getting rid of it, and you do that through secretion. So secretion is like the last minute, you didn't get it in time on the filtration, but you don't want to wait until it cycles through the whole body again. So secretion is kind of like your last ditch attempt. And you'll notice it's not the same things. Okay. The things that can go through filtration cannot always go through secretion. Does that make sense? So all the things that can be filtered cannot necessarily be secreted. And what determines the difference is the size. Okay. Larger things like glucose, protein, those things can go through filtration, but it can't go through secretion because it's too large. It can't move through this membrane. Okay. where the filtration slit formed by the glomerular capsule, the fenestrated endothelium formed by the blood vessel, it can work through there, but it can't work through the capillary, the peritubular capillary into the renal tubules. Does that make sense? Only limited things can be secreted. Okay? Questions?
So because this was a test question that I had to think about, and thankfully I got it correct. But with the secretion, that is from the peritubular capillaries back into the tubule so that it can exit out of the system through the collecting ducts, correct?
Correct. That's right.
Okay. And then for your reabsorption, that's just the opposite. So it would be
Exactly.
okay, gotcha.
Yep.
I had one quick question because this might have been in the text, but I missed it. Does the secretion require, because it's going the opposite way, does that require any particular like active transport to happen, or no, it's just like the
No, it still requires active transport.
okay, I thought so.
Yep.
I just wanted to make sure of that.
Yep. Yep. Okay. All right. But luckily, those areas already have the mitochondria, so it's all good for the pumps to kind of move things back and forth. Okay. All right. So let's see. What else do I need to add to here? So that's it. Okay. Let's quickly go into this. So this is the filtration membrane. So we know that the filtration membrane occurs at the renal corpuscle. Here's a new term. The renal corpuscle just means the connection between the glomerulus and the glomerular capsule. Okay, so the renal corpuscle refers to the connection between the glomerulus and the glomerular capsule. So this is what it looks like. So you can see that from the glomerulus, so this right here, that's the glomerulus. And we know that the glomerulus is a network of capillaries, right? And just as a review, capillaries in general have one layer called the endothelium. You'll notice though that this endothelium is specialized because it has these little windows right here. And the German word for windows is Fenestra. So that's why this particular endothelium of the glomerulus is referred to as the fenestrated endothelium. Okay, then you have the basement layer, the basal lamina that connects the glomerulus to the glomerular capsule. Then you have the glomerular capsule. The glomerular capsule consists of the simple cuboidal epithelium. It has these specialized cells called podocytes found in the visceral layer, in other words, the inside layer. Because remember, this is the parietal layer and this is the visceral layer. So in the visceral layer, they have these podocytes that have these extensions right here that form what's called filtration slits. So blood is going to move from the glomerulus past the membrane, past the filtration slits into the glomerular capsule. Then it's going to continue onward to the PCT, to the DCT, and so on. Okay, so this is the filtration membrane. The filtration membrane consists of these three parts: the fenestrated endothelium, the basal lamina, and then the podocyte of the visceral layer of the glomerular capsule. Okay, so these three parts. Okay. All right. So what we need to do then is look at this one right here. So Aaron's question was on the urine formation process. Urine formation. And we've actually already complete segments of it. Okay, So in the urine formation process, the first one is glomerular filtration. Glomerular filtration allows for the majority of substances in plasma to go from the glomerulus into the glomerular capsule. Right, we just finished talking about that filtration membrane right here between these three parts, the fenestrated endothelium, the basal lamina, and then the podocytes of the visceral layer of the glomerular capsule. Okay, so things are going to enter, and that's filtration. Okay, I don't have time to go through that, but you definitely, I think I did in my lecture. Make sure you understand what glomerular filtrate rate is, what it is, and so on. Okay. Then once substance goes in, it can move from the renal tubules into the back into the blood vessel. And when it moves back into the blood vessel, it's called tubular reabsorption. So that's the second step. Tubular reabsorption. Okay. Now it's gonna continue onward and then end up in urine. Okay. Let's say your body though has a little bit too much of something and still wants to get rid of certain things. You can see that from the blood vessel, it can go back into the renal tubules, this one right here, in this direction, in which that's called tubular secretion. Okay. So tubular reabsorption is when it's going from the renal tubule into the capillary. Tubular secretion is when it's going from the capillary back into the renal tubules. Okay, so you can expect test questions. For example, in tubular secretion, what was the fluid and what has it become? So in tubular secretion, what was the fluid called and what has it become? What was the fluid in tubular secretion and what has it become?
Filtrate in urine.
Not quite. So here's secretion right here. So where was it originally and where did it go? It was originally in where?
Capillary.
Yes.
Capillaries?
So if it was in the capillary, we call that plasma. And now it goes into where?
Tubule.
Yes, the renal tubule. So then we call it filtrate. It's not called urine until it gets the collecting duct. So until it gets into the collecting duct, that's when it's called urine. Okay, how about the tubular reabsorption? What was the fluid and what has it become? In tubular reabsorption, what was the fluid called and what has it become?
It was filtrate and becomes plasma.
Perfect, you got it. And that has been a test question in the past. How about glomerular filtration? What was it? What has it become? Come on, you can do this. What was it? What has it become?
In the glomerulus, it was plasma, and the capsule is filtrate.
Perfect. You got it. Okay. All right. So that is that. Skipping a lot of things because we're out of time. But that's pretty much it. The endocrine system, mostly memorization. Do that chart. Make sure you know where hormones are secreted, what's their target, what's their action. So this morning, we had a chance to go through the urinary system. Before I do that, I just want to review some logistics. The lecture exam has 100 questions, 50 on the old materials, and it covers all the chapters that were covered in exam 1, 2, and 3, and they're all multiple choice, which means there's about 3 to 4 questions per old chapter, which is why I really recommend you focusing on the new materials, because they're going to be a lot more detailed. Because within the last 3 chapters, the urinary, endo, and reproductive, I have 50 questions to spread out amongst those. So think of the last 50 questions as your 4th exam. So 40 questions will be multiple choice, 10 will be fill in the blank, short answers. And then for lab is 50 questions, about 25 on the old materials and about 25 on the new materials. And all 50 questions, you have to type in the term and then spelling counts and just some things to make sure that you add into your identification would be if there's a pair of them, make sure you indicate right or left. If it's a blood vessel, indicate whether it's an artery or a vein or capillary. And then this morning, someone had a question about if I were to point an arrow and that you're not quite sure what I'm pointing to, always go with the specific and then add the generic. So for example, here's the liver. I have an arrow right there. What you could do to kind of cover all your bases would be right lobe of the liver. All right, and plus one big thing to pay attention to is the question themselves. Usually they are very clear about what I'm asking for. So for example, like what chamber is this? What specific structure is this? Or what segment is this? Or what organ is this? So on, okay? And then make sure you use respondents lockdown. And unlike the other 3 exams, I'm gonna have to go through and watch every environmental scan. So please, please, please make sure you're doing it correctly. How do you know you're doing it correctly? I attached a video in my emails of pretty much all of your exams of what an environmental scan should look like, okay? And one major difference is that your final exam is actually from Friday to Sunday, not Saturday to Monday. So you do not have until Monday, okay? So you have to take the exam by 11 p.m. Central Standard Time on Sunday. I think those were the main things. Did I forget anything? Or do you have any questions on the exam?
I think you got it covered from like the same one this morning, Professor.
Awesome, thank you. Okay, so I'm gonna jump to reproductive. Okay, so like I said, we're going to do the female reproductive system first. It's just more difficult, okay? So to understand, you have to make sure you understand the following terms. Gametes. What are gametes?
Sex cells produced by the gonads.
Yeah. So there's
Sperm for male, ovum or eggs for female.
good. What characteristics do they have? How many chromosomes?
Is it 23 each?
23 total.
Okay.
So 23 total chromosomes. And what's fertilization?
Is that when the sperm joins with the egg?
Yep. The sperm is able to break through the wall. Yep. So the sperm penetrates the egg. Okay. And what's a zygote?
Fertilized egg.
What is it?
Fertilized egg.
Yes. And combination of chromosomes. So how many chromosomes would a zygote have total?
23. Oh, no, it's 46.
There you go,
Right.
46.
46.
23 from the egg and 23 from the sperm, okay?
Right.
All right, then what we say we've been saying chromosomes. What exactly is a chromosome?
Genetic map. I don't know.
Yep. So DNA, genetic information, okay? So we have a couple of ways in which we describe chromosomes, right? 46, 23, autosomes, sex chromosomes. Make sure you know the difference between them, okay? So a cell can be considered to have 46 chromosomes, can also be written as 2N. So 46 can be written as 2N. And what's another way of saying 46 chromosomes?
Professor, isn't that like haploid if it's 2N?
Nope, you actually have it
Diploid.
reversed. Diploid.
Diploid?
Yeah. So
I
that's
remember haploid. I remember haploid because half translates sounds like half, and that's usually one.
exactly. That's right, Jamie. So half is half. Yep. Now, let's just leave it that for now. Okay, so then 23 total chromosomes would then be N, and that's a haploid. Okay. Within these 23 or 46 chromosomes, we have the term autosomes and sex chromosomes. So
Can I ask a question?
yeah, go ahead.
I'm sorry. When is it 1N versus 2N? Just a point of clarification on that.
When is it what?
Is haploid ever is it written as 1N also, or is it just N?
Yeah, you can have it as 1N or just N. It's the same thing, right?
Okay.
One times something is itself. Yep.
Okay, I just want to make sure I had it. Thank you.
Yeah, definitely. Always clarify. So even though it's 46 chromosomes or 23 chromosomes, particularly with the 46 is actually pairs. Okay. So for example, chromosome 1, chromosome 1, chromosome 2, chromosome 2, all the way down to chromosome 23. Now the 23rd chromosome does not necessarily have to be a pair. So the 23rd chromosome can have the combination of X or XY. So when they are identical to each other, chromosome 1 through chromosome 22, they are referred to as autosomes because they are identical to each other. So autosomes means that they are identical pairs. Okay, chromosome 1, chromosome 2, 2, so on. The 23rd pair is referred to as the sex chromosome pair because they may not be identical. Okay, so you can either have a combination of XX, which gives you what gender?
XX is female, XY is male.
Perfect. Okay, all right. Where 23 chromosome is just chromosome 1, a single chromosome 1 through 23. Okay. So that's the difference between a diploid and a haploid. And within the differences, what do the chromosomes look like? Okay. So then we move on to cell division. There are two main types of cell division. There's mitosis with a T and meiosis. Okay. So mitosis and meiosis. Mitosis is cell division of what?
Zygote or any other somatic cells?
Yep. So these are all cells except gametes. So mitosis is division of all cells except for gametes, where meiosis is cell division of only gametes. Okay. All right.
I remember gametes and meiosis because they both have an E in them.
There you go. I love the mnemonics and the memory clues that you have. They are very helpful. Okay. Now, in terms of mitosis, you're going to see that today or now when we talk about the female reproductive system, you're going to see that in mitosis, they're going to produce identical cells. Produce identical cells. Okay, So in mitosis, they're going to produce identical cells. So one cell that's 2N will result in what?
2N.
2 cells that are 2N. Okay, not 3. Okay, because it's not like this 2N gave birth to 2, right? No, this cell that's the original 2N split in half and became 2. So from 1, you end up with 2, not 3. Okay, so from 1, it splits, it copies, and then it splits, and then produces 2. And these 2 new cells are identical to each other, as well as they are identical to what they originally were. Okay, so it's the same cells. That's why your skin cells are exactly identical to each other. Because your skin cells would be an example of a cell that is highly mitotic. Okay, where in meiosis is a way of narrowing down chromosomes. Okay, so from 1 to N, it's going to eventually result in 4 cells that are N each. So it's going to copy and then split. Okay, so there's still a copy and divide, but it does so that it ends up in 4 cells, and each of those 4 cells have 1 N. Okay, now if we're talking about the male reproductive system, then they will definitely result in 4 Ns, viable 4 Ns. In females, because females is about quality over quantity, she's just gonna have 1 N altogether, and these 3 will break down. It's just a method of getting rid of the extra chromosome. Okay, so that's meiosis. Okay, and we're going to see that applied to the female and male reproductive systems. Okay, make sure you're able to describe the functions of the female reproductive system. Make sure you're able to know the organs and their functions. What's the ovary, what's the uterine tube, what's the uterus, the vagina, and other external female organs. And this is the one that we're going to focus on, is oogenesis. Okay. Now, oogenesis is just one part. The reason why this is so complicated is because the female reproductive cycle actually involves the ovarian cycle and the menstrual cycle. Okay, so it's quite complicated because it has 3 events that overlap each other. All right. Questions? Okay. So here is the external structures. So what would you call this? Number 1?
Ovary.
Ovaries.
Yep, right and left ovary. And then it becomes number 2. And number 2 has 3 parts. 2A, 2B, and 2C.
Membrane.
Ovarian ligament and the uterine.
No. So we're just on 2. So what's number 2?
Uterine tube.
Okay.
And then 2C is fallopian tube.
No, the uterine tube used to be called the fallopian tube.
Oh, okay. So 2 is the fallopian tube.
2, yep. 2
Okay.
is the fallopian tube, yep. So let's do 2A. 2A is the easier one. So 2A right here is the fimbriae.
Oh.
What's its job?
To help move the eggs.
Yep, it sweeps the
the
egg inward. Exactly. Otherwise, the egg is actually released into this space right here. And if a sperm comes along and fertilizes that egg, what could happen? In the pelvic cavity.
Endometrial pregnancy, or what it's called?
Yep, ectopic pregnancy.
The topic is it? Yeah.
Yep, okay. So that's the fimbriae. Fimbriae sweeps the egg in once it's released. What's 2B? And this image is not quite right because it 2B swells up a little bit.
Oh, yeah. Crap.
That's the ampulla. And then 2C is what connects to the uterus. So what would 2C be? Starts with an I. That would be the isthmus. Okay, so that's the uterine tube. And now specialized, well, we'll actually come back and fill this out more. Ovary. So ovary is where the egg is produced and secreted. The uterine tube, what's specialized about that is that that's where fertilization must occur. Why must fertilization occur as it travels through the uterine tube?
So I can get to the uterus.
Nope. Why doesn't it get fertilized in the uterus?
Well, I mean, isn't it it attaches to the uterine wall so it can grow, but then on the process, don't it doesn't go to like like maturity in a way?
No. Others? What do you think? Why does the uterine tube have to be the site of fertilization?
Because there's only one,
Because there's what?
only one egg. I don't know.
There is only one egg. That is true. Well, sometimes there's two, but mostly there's one egg. But that doesn't tell us why does number 2 have to be the site of fertilization? Why can't the egg get fertilized in the uterus? That's where it gets implanted. So why can't it get fertilized there? Eggs are only viable for about 1 to 2 days. Okay, eggs are only viable for about 1 to 2 days. It's kind of scary, right? I mean, like, human population is on the rise, so you think that the window of opportunity to get pregnant is much wider, but no. Once the egg is released, it is only viable for about 1 to 2 days. And it takes about anywhere from 3 to 5 days to make its way down the uterine tube. Okay, so that's why the uterine tube has to be the site of fertilization, because the egg is only viable for 1 to 2 days. Okay, then you have number 3 right here.
Well, can I ask you a question on that?
Yeah.
Well, if that's the case, does that mean that the sperm and their movement is much faster, and because of the flagella, they can move a lot faster, and they're able to get in there and do the fertilization? Is that the reason why? Because obviously they move fast, and compared to the egg.
Yeah. Yep. That's right.
So there's
They
nothing
have to be
to move.
they
There's
have to
nothing
be
to
able to
move.
swim up.
Right.
Yep. They have to go
Sorry.
all the way through to the uterine tube to be able to fertilize. So that's why they have to be very motile. Okay. So
Okay. Well, how long? But can I ask one more question? But how
Yeah.
long would it be from when it ejaculates to get there? I mean, how long would that be?
Oh,
Typically?
it doesn't take long. No, it doesn't take long at all. It depends on the motility of the egg. I'm sorry, of the sperm, but it can make its way up like right away.
Okay.
It doesn't take long to travel through. Yeah, because it's moving by itself, where the egg is just like moving with the flow of as things move through. There's a difference.
Right.
So because of the
Okay.
flagella,
Right.
the sperm is able to make it up like within. Don't quote me on this, but I believe it's within like hours.
Okay. All right.
Yeah.
Just a general idea. Okay. Good. Thank you.
Yes. So how about number 3? What's number 3?
Uterus.
Yay, thank you. Uterus. The uterus has 3 parts, A, B, and C. What's 3A, 3B, and 3C?
3A is the fundus.
Yep.
Fundic. Is it fundic? Fundus? What? Give me the.
You can use either term, fundic or fundus.
Okay.
Technically, it's the fundus.
Okay.
Yes. How about
Thank you.
3B?
Just
It's the
uterus,
body
right?
of the uterus.
The
And
body of the
3C is the
uterus.
cervix.
And C is the cervix, the neck. Okay. So just think, remember, you have cervical bones, you have thoracic bones, you have lumbar bones, the vertebrae. So because this is the neck of the uterus, we use the term cervix. Okay, because the term cervical or cervix means neck. Okay. All right. Now I'm going to take a little detour and talk about the layers. So you have this layer. Oh, white might not be the best color. Let's choose green. So you have this outer layer, this middle layer, and then this inner layer. Okay, so the uterus has 3 layers. Which would you prefer, superficial to deep or deep to superficial?
Superficial to deep.
Superficial to deep it is. Let's do okay, let's do Roman numeral 1, Roman numeral 2, Roman numeral 3, because you want to do superficial to deep. What's the outermost layer?
Parametrium.
Is it myometrium?
I have a couple different answers. What do you all think?
Parametrium.
Parametrium it is. Parametrium. Okay. How about 2? Aaron, what would 2 be then?
Myometrium?
That's the myo. Myo means muscle, okay? So myometrium, because it's the muscle of the uterus. And then 3 would be
Endometrium.
endometrium. Now the endometrium has 2 sublayers. So the endometrium has 2 sublayers. What are the 2 sublayers of the endometrium? You have the stratum what?
Functional.
Yes. So
Functionalis.
stratum functionalis and stratum,
Basalis.
yep,
Is it basalis or basalis?
basalis.
Okay, basalis.
All right. Then you have this next structure. So 4. What's number 4?
Vagina.
Yes,
Canal.
the birth canal.
Yeah.
Yep. Okay, so those are the external structures.
Okay, question for clarification. If we said vagina, would that count, or do we need to say vagina canal?
Vagina is fine.
Okay,
Vagina is actually the technical term. It's kind of like the layman's term is the birth canal. Or you can say vaginal canal. Yep. Okay. So now we're going to go to the request to color. So that's going to be oogenesis. Okay. And I'm trying to see which, yeah, let's do this way. Because sometimes it's easier to see the big picture and go into the smaller details. Sometimes it's easiest to go through the small details and then align them. I think for this, the oogenesis, it's easier to do the smaller details and then align them than to see the big picture first. Okay. All right. So we're going to go small details to alignment. All right. So the first thing that we're going to talk about is oogenesis or oogenesis. You can pronounce it either way. Okay. And essentially it means the development of the ovum. Okay. Now, the female reproductive cycle is very interesting because this actually starts in the female's fetal development. So when the female is still developing in her mother's womb, that's when oogenesis begins. very different from spermatogenesis in males, which begins at puberty. Okay. So the oogenesis actually begins at fetal development. So when the female is still in her mother's womb. Okay. Starts off with oogonia. And oogonia is the stem cells. Okay, so here's an oogonia. And you can see that oogonias have how many chromosomes total? How many chromosomes total? I should show it in. Not 23, good guess though, Liz. 46, because it's 2N. Do you see the 2N right there? So it has 46 chromosomes. Okay, so in mitosis involves a couple things. Okay, so in mitosis, it's going to be a copy and divide. Pretty straightforward. It copies and then it divides. So let's go through this step by step. So if it has 46, when it copies, how many chromosomes will it have? So times 2. $92, very good. Thank you, Nelly. So it has 92. So it's copied. So now inside that cell, it's going to have 92 chromosomes. You can think of it as 4N even if you like. Then it's going to divide. It's going to split right in the middle and result in this cell and a second cell. Okay. So one more time. You start out with an oogonia. It undergoes mitosis. It copies its chromosome from 46 to 92, and then it divides. It splits right in half, resulting in 2 new cells. Each of these 2 new cells have 2N in them. And remember, mitosis produces what type of cells? Different or identical? Identical. A good analogy that I give students is that here you have, let's say I gave you a piece of paper, right? I gave you a piece of paper with my picture on it. And I told you go to photocopy it. You put it into the machine, you photocopy, you make 1 copy of it. It's identical. You still have the original, and now you have this. Okay, so same thing here. That's why it results in 2 and not 3. You don't have the original, and then you don't have the 2 new ones, right? The original divided into to make the 2 new ones. So all together from 1 cell, 1 stem cell, you end up with 2 daughter cells. We call them daughter cells. Okay, and they are identical to each other, and they're identical to the parent cell. Okay, so from 1 you end up with 2. Now one of those 2, okay, becomes what's called the primary oocyte. One of them becomes the primary oocyte. The other one is going to go back and repeat this process and continue to divide. Okay, so one of those new cells that results from mitosis goes on to become a primary oocyte, where the other one stays as an oogonia, goes back through mitosis and repeats this process so that you continuously make multiple copies. Okay, all of this is happening during when? This is mitosis, but when during this female's life cycle? or lifespan is this occurring?
At birth?
No, during fetal
Before birth.
development. This is even before she's born. That's how crazy this is. So a female is born with all the eggs she will have. That does mean, however, that as she ages, what else ages with her?
Eggs.
Yes. Where males, they're producing sperm at puberty until they die. So their sperm is newly produced. Okay? And that's why men can have children old. Where females, not so much, because chromosomal defects start to happen when the eggs get too old. Okay? All right. So that's the first step. So far we just covered the first step. Okay? Which is during fetal development, the oogonia undergoes mitosis and makes millions of cells. Some become primary oocytes. Okay? That's all we talked about so far. Questions before we move on? Okay? Now, meiosis in general has 2 steps. Okay? So you have meiosis 1 and meiosis 2. Okay? So you have meiosis 1 and you have meiosis 2. Meiosis 1 is a simple divide. How's it different from mitosis? How is meiosis 1, which only goes through divide, different from mitosis? Yeah, it's only dividing the gamete. What did it not do before it divided? You're right, Michelle. It only there you go. It doesn't copy, because overall the purpose of meiosis is not to produce more cells necessarily, but it's a way to narrow down the chromosomes. Okay, and that's why meiosis works with just gametes. Okay, so in meiosis 1, it's a pure and simple divide. It's as a means of getting down to just one end. Then once you're down to one end, meiosis 2, that's when you make more of those cells. So in meiosis 2, now we're going back to copy and divide. Okay, but let's go back to our drawing over here. Okay, so during fetal development, the oogonia is going to undergo mitosis, which produces a primary oocyte and an oogonia. That oogonia repeats that cycle to produce millions of primary oocyte. Okay, now those primary oocytes begin meiosis 1. So first meiosis, that's meiosis 1. Remind me what happens in meiosis 1 again?
Copying.
What is it?
You copying?
Careful. What happens in meiosis 1? It divides.
Yeah.
No copy. It just divides, which is why from one primary oocyte, that's a 2N, it divides and becomes 2 cells that are 1N each, because it didn't copy initially. It just split in half. You have 2N. When you split in half, one goes to this side, one goes to the other side. Okay? However, this process actually doesn't even finish, because right here, doot, 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 it stops. So at birth, this stops. It undergoes cell arrest. It stops. So it actually starts meiosis 1. It doesn't complete it. Okay. It just plain stops. Okay. Then, so a female is born with all the primary oocytes that she will have, that has begun meiosis 1, but has not completed it. Okay. So true or false? A female at birth will have a combination of primary oocytes and secondary oocytes. True or false? A female at birth, there you go, false, because it never completes meiosis 1. Okay. However, at puberty, all the hormones start going crazy, right? Particularly FSH, follicle stimulating hormone. The follicle stimulating hormone at puberty is going to cause the completion of meiosis 1. Okay, so each month, a handful of the primary oocytes will finish meiosis 1 and become a secondary oocyte. Okay, now in females only, not in males, but in females only, when it divides, it does not divide evenly in terms of the cytoplasm. Okay, one of these, this one will say, let's see, let's do this, insert, shape, let's get rid of this. Yeah, and then like so. Okay, perfect. Okay, so technically, do this. It's gonna, when it undergoes it, when it finishes meiosis 1, it will actually produce like this. Okay, one of the new cells, the new gametes, will have all of this, all of the good stuff, all of the mitochondria, all the organelles, all the cytoplasm, everything. Just half of the chromosome though. Okay, the other one will be teeny little bit because it's a way of getting rid of the extra chromosome. That one will eventually undergo atresia and disintegrate. And that's why we call it a polar body. Will disintegrate. Disintegrate. Or a form of atresia. Okay. So really, at the end of meiosis 1, even though it results in 2 cells that are 1 end each, really, how many do you have though? Just 1, exactly. Okay, and that one is called what? Not yet. Yes, a secondary oocyte. And it turns out that it's this secondary oocyte right here that is ovulated. each month. Okay, so one cell total, more specifically, one secondary oocyte is released in usually per month. Sometimes both sides release one egg, but usually one side releases one egg per month. Okay, and it stops there. It stops at meiosis 1, after completing meiosis 1. Okay, now there is a specific condition that will allow, oh, I shouldn't use that color. Let's use, let's do green. In order for meiosis 2 to occur, fertilization must happen. So in order for meiosis to occur, in order for meiosis 2 to occur, fertilization must happen. Fertilization is required. Okay, so fertilization is required for meiosis 2 to occur. If fertilization does not happen, in other words, if the sperm never comes and penetrates the egg, then that secondary oocyte is just going to be expelled through what process? So that secondary oocyte is going to be swept up by the fimbriae, go to the uterine tube, to the uterus, and then what happens? Yes, it's expelled through menstruation. Exactly. And that's the end of that story. If fertilization does happen, then we worry about what comes next. Okay, so if fertilization does happen, okay, so let's show that happening. Let's do here. Let's insert. I want to change that again. White. Draw. And here. Okay. So if fertilization does happen, let's make the egg purple. I'm sorry, the sperm purple. Okay, so if the sperm is able to penetrate the egg, the minute it penetrates the egg, that egg or that secondary oocyte, okay, is going to complete meiosis. It's going to start meiosis 2. Okay, why? Because that's just what happens in nature. In nature, you have to have 2 parts of meiosis. Do you need it to happen? No, because you're already at N and N, right? You're already at the chromosome level that can combine to produce a zygote that's 46 or 2N, okay? But it's going to do it because that's just what happens. You know, it has to complete meiosis, and meiosis has 2 steps, okay? So when it completes meiosis 2, okay, so the minute that sperm penetrates that egg, it's going to complete it. Did it need it to? No, not really. But in the meantime, let's just take a side note right here. Let me use a different color. Let's do a side note. So the minute that egg penetrates, I should do it this way. There we go. Okay, so the minute that sperm penetrates the egg, the wall of the egg, the secondary oocyte is going to undergo meiosis 2. And you know that in meiosis 2, it's going to copy and then divide. So it's going to go from 1N, it's going to copy and become 2N. Okay. And then it's going to divide. Okay. That one N, that's just a way of getting rid of that extra chromosome, is also a polar body and also undergoes atresia. This one N right here is going to combine with the sperm one N at this point, since it finishes meiosis 2, and then it becomes what? So after that secondary oocyte gets penetrated by the sperm, it's going to finish meiosis 2, and it's going to combine the genetic information with the sperm and become a what? A zygote. Thank you. At this point, this zygote is going to divide, copy and divide through what process? Mitosis or meiosis? Yes, mitosis. Perfect. Until it becomes a fetus. Okay? How was that? Questions? That's kind of cool, isn't it? I mean, I think it's pretty dang cool. Okay. So truly, females are about quality over quantity. Okay? So that's oogenesis. It starts off with mitosis during fetal development, starts meiosis 1, but doesn't complete it until puberty. It finishes meiosis 1, gets expelled through ovulation. If it's fertilized, it continues on with meiosis 2, and then join with the chromosomes from the sperm and become a zygote. If not, the secondary oocyte it gets expelled through menstruation. Okay, questions? Okay, that's part 1. We have 2 more parts to go through. Okay. All right. So far, what we did is that we've covered this side right here. We covered the left side. So we finished that side. Now we're going to go look at follicular genesis. Okay. All of this is within the ovarian cycle. So in the ovarian cycle, you have to follow 2 things: the development of the egg and the development of the follicle. We just covered the development of the egg. So now we're going to cover the development of the follicle. Okay. So it's nice that we covered the egg array, because now you can just kind of compare and contrast. Okay. So you can see that inside you have the egg, but around the egg you have a follicle. Now the follicle is produced, I don't think let me erase this first. Hold on. There you go. Didn't quite finish erasing it from the last time I used this slide. All right. Okay, so you can see that you still have the egg, but now it's more complicated than that. So it's like we're doing layers at a time. Okay, so we just finished the one layer of the whole story. Now we're going to talk about the other layer that happens at the same time. Okay, all right, so you still have the oogonia, and it turns out that the oogonia is surrounded by a follicle, specifically the primordial follicle. The primordial follicle surrounds the oogonia during the female's fetal development. So what is a follicle? A follicle is consistent of these cells right here. So the follicle itself is made up of follicular cells. Okay, so the primordial follicle is made up of these follicular cells. Okay, questions? All right. Okay. Where are we? Okay. Now, during fetal development, this primordial follicle are made up of these follicular cells do not change. Okay, so there's no change during fetal development. Okay, so during fetal development, true or false? There's a change to both the oogonia and the follicle. False, exactly. So there's only change to the oogonia, but not the follicle. Okay, so that's good. So we don't have to worry about the follicle until puberty, adolescent. So at puberty, what happens again that triggers this whole process to begin? No. FSH. Now it makes sense, right? FSH stands for follicle stimulating hormone. Yes. Well, sorta. It starts the process, and you'll see how it is connected to each other. Okay. So the follicle stimulating hormone, it's named follicle stimulating hormone because why? It stimulates the follicle. It's going to cause the primordial follicle to change and become the primary follicle. Okay, so FSH causes the change from the primordial follicle to the primary follicle. So where the primordial follicle consists of follicular cells, the primary follicle consists of these granulosa cells. Okay. What are some other differences that you notice between the primordial follicle to the primary follicle, besides the fact that the cells are different? You know, how about in the tissues? What would you call the primordial follicle? Is it simple or is it stratified? Simple, where the primary follicle starts to become what? Let me zoom in a little bit. Yeah, exactly. Okay, so you will find that FSH is going to stimulate about 20 to 25 primordial follicles to develop into a primary follicle. Okay, so it starts off as simple squamous, and when it becomes the primary follicle, it becomes simple cuboidal. But the primary follicle is going to become stratified. When it completely becomes stratified, that's when it transitions from the primary follicle to the secondary follicle. Okay, so you can see that it begins from the primary follicle. Initially it retains the simple cuboidal nature, but then it's going to start to develop and it's going to become stratified, to which then is now referred to as secondary follicle. Okay, so there's still granulosa cells, but it becomes fully stratified when it reaches the secondary follicle stage, and it forms the zona pellucida, as well as the stelca folliculi. Okay, the important part is the zona pellucida. The zona pellucida is this layer right here. I'm going to highlight it in green. Okay, so the zona pellucida is right here. It forms a space between the egg and the follicle. And that zona pellucida is going to continue to grow and become what's called the antrum. And now when it forms an antrum, that's when we call it the tertiary follicle. And in some books, it's just referred to as kind of the mature follicle. Okay. The importance of that antrum is because it creates pressure so that when it switches from the follicle stimulating hormone to the luteinizing hormone, it's going to cause that antrum to rupture and thus release the egg through a process of ovulation. Okay. So let's go through that one more time. Okay. You start off as the primordial follicle with simple squamous epithelium. Okay. And that's just stays the same during fetal development. Then at puberty, FSH is going to stimulate about 20 to 25 primordial follicles. So not everything a female is born with, but about 20, 25 of these primordial follicles develop and become the primary follicles. The difference is that it starts off as simple, but it starts to become stratified, as well as it changes shape to cuboidal. Okay. And it's going to become stratified to a point where then it's called a secondary follicle. So at this point, it is fully stratified and cuboidal. Okay. Now the secondary follicle is unique in that it has the zona pellucida, an area between the egg and the follicle itself. And that secondary follicle is going to become a mature follicle, and we'll just put tertiary here so that you can associate the term together. Okay. So what's going to happen here is that LH, at this point, LH takes over, and LH is going to cause the zona pellucida, which is now called the antrum, to rupture. And when it ruptures, it releases the egg. Okay. Now, the follicle that remains back, is now referred to as the corpus luteum. And the corpus luteum has a responsibility as well. So the corpus luteum is responsible for producing progesterone. We're going to figure out why that is important in a little bit. Okay. So you have two hormones that play a role here. FSH starts the process, and eventually it's going to switch over to LH, the luteinizing hormone. Okay, so luteinizing hormone has an effect on the corpus luteum. As long as the luteinizing hormone is around, the corpus luteum is also going to stay around. The corpus luteum itself produces its own hormone called progesterone, and we're going to talk about that in a little bit. Okay, so that's the follicular genesis. You have primordial follicle at during fetal development that becomes the primary follicle, at puberty. And then the primary follicle, through stimulation of FSH, develops into a secondary follicle, to a tertiary / mature follicle, and then it's going to release the egg. And that's how the egg is released. Okay. So let's look at this here. So far we've just covered the ovarian cycle. So you can see that in the ovarian cycle here, okay, during the follicular phase, which is controlled by FSH, the follicles change. And as the follicles change, they cause the egg to develop and then to be ovulated. Okay. Then at ovulation, what actually causes ovulation to happen is that FSH loses control to LH, the luteinizing hormone. So as FSH goes down, LH goes up. Okay. When LH goes up, it leads to ovulation, and LH then controls the corpus luteum, controls the second half of the cycle. Okay. All right, now to add a little bit more difficulty to it, we also have the roles of 2 additional hormones. You've got estrogen right here and progesterone, and to some extent remaining estrogen. Okay, we already talked about progesterone, right? We said that progesterone is produced by the corpus luteum. Okay, the progesterone, you'll notice that progesterone causes the uterine lining to become thicker. That's the role of the progesterone, because now your body has released, the female body has released the egg, and it's preparing for pregnancy in case fertilization happens. So to prepare for that, regardless of whether it happens or not, the corpus luteum is going to produce the progesterone, and progesterone is going to cause the lining to get thick. Okay, and become thick with blood so that it can nourish if fertilization does happen. Okay, what we haven't talked about yet is the role of estrogen. Okay, so estrogen, when the follicle is developing, the follicles are going to produce estrogen initially. Okay, specifically the antrum. Okay, so in the antrum, or in that zona pellucida, okay, estrogen is going to be converted. Okay, so estrogen is going to be made essentially from the chemicals that you have in the antrum. Okay, so estrogen is going to result, and estrogen actually is going to be what starts the uterine lining it's going to start the uterine lining to develop. Okay, so that's when the stratum bacilli, or bacilli, whichever way you want to pronounce it, starts to form the stratum functionalis, and that's due to the estrogen. Okay. Now, at this process where the antrum starts to produce the estrogen, which then causes the lining of the uterus to form, it actually has a negative effect on FSH. Okay, it's going to reduce receptors for FSH. Okay, it's going to reduce the receptors for FSH, so that of those 20 to 25 follicles / egg that goes through this process, whichever one matures the fastest, okay, is going to continue to develop, and all the others start to die off. Okay, so it's really selecting for the best of that crop of 20 to 25 that gets stimulated by FSH per month. Okay, so the one that reaches maturity first, you can think of it as it turns off all the others. Okay, so then the others never become mature, and they start to die off, okay, or disintegrate. Okay, so that means that the one that made it to become a mature follicle. That's the one that's going to release the egg in ovulation in response to LH. The egg is going to move through the uterine tube and see if it gets fertilized or not. If not, it's just going to continue down and then get flushed out through menstruation. If it does get fertilized, okay, then it's going to implant itself along this newly made lining made by estrogen, maintained by progesterone. Okay, it's going to attach itself to that lining. Okay, now here's the thing. Luteinizing hormone, okay, is going to cause the decrease because the antrom's gone, right? So you're going to start to see a decrease in estrogen and increase in progesterone. Estrogen is still there, but progesterone takes over. Okay, now the luteinizing hormone is going to decrease. Now, if the luteinizing hormone decreases, what happens to the corpus luteum? Because it's the luteinizing hormone. Yeah, exactly. The corpus luteum is going to shrink and die off. Yep, it becomes albicans. Very good. Okay. Now, if the corpus luteum shrinks, dies off, or becomes the albicans, what stopped being produced? So you have to backtrack. What is the corpus luteum responsible for producing? Progesterone, exactly. Progesterone, okay? And if progesterone disappears, what happens to the lining? The lining can no longer be maintained, right? So what happens next? Question, hold on, let me see. Menstruation, exactly. Jamie, I have to leave group now, but I have one last question regarding the chapters we had in exam 1, 2, 3. Are there questions from the exams? Not necessarily, no. Nope, they will be similar, but they're not exactly the same. Okay, like you said that this morning. So the lab is going to be the same because the lab is the same images, but the lecture will not be the same. Okay. All right. So yeah, so menstruation is going to happen, right? So what happens if the egg is fertilized? So what happens then is if the egg is fertilized, so here's the egg, oops. So if the egg is fertilized, that zygote is going to produce what's called the human corticotropic hormone. Okay, HCG, human corticotropic hormone. So the HCG, or what we call the pregnancy hormone, so that's why when you take pregnancy tests, it's measuring for the HCG. So the HCG replaces the luteinizing hormone. Even though luteinizing hormone is not there, it replaces it by maintaining the corpus luteum. And if it maintains the corpus luteum, it's going to continue the production of progesterone so that the progesterone maintains that lining for the 9 months that the woman is pregnant. Okay, so that's what happens, kind of like the whole story. So before I end, I'm going to show you, there is I'm not sure if you have access to it, but there's this really cool cognitive map that I created for this class. I'm going to share it with you. Zoom, more, share screen. This one. Okay, can you see this okay? Can you see the cognitive map?
Yes.
Okay, so this cognitive map kind of goes through exactly what we just covered, the oogenesis, the follicular genesis, and the uterine wall by day, by cycle. So it kind of divides it up to everything you need to know. Okay, so this is a really good study tool. What you can do is that you can kind of erase it, try to see if you can write it, figure out which is which. Okay.